Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
You know, right now, inside every single one of your cells, there's this microscopic factory that's just hard at work. And its only job is to build the very letters that write your genetic code, completely from scratch. Today, we're going to take a tour of that assembly line. I mean, it's almost impossible to really wrap your head around that scale. We're talking trillions of individual cells, and each one is like its own little bustling city, full of perfectly coordinated chemical reactions. It's an entire universe working inside of you at this very moment. And deep inside every one of those cells, at the very heart of it all, is the blueprint for life, DNA and RNA. You can think of it as an alphabet with just 4 letters. But here's the really wild part. We don't get these letters from the food we eat. Our bodies have to build them, literally, atom by atom. So what we're talking about is a process called de novo pyrimidine synthesis. De novo just means from the beginning. And it's not just some boring series of chemical reactions, it's the actual story of how life builds itself from the ground up. So let's dive in.
All right, so let's zoom in a little. We're going to focus on one specific family of these letters, the pyrimidines. You've probably heard their names, cytosine, thymine, and uracil. These aren't just abstract things on a chart. They are the absolute essential building blocks for the DNA and RNA that make you, well, you.
So how does the body actually do it? Well, it's this incredibly elegant 8-step process. And really, the best way to picture it in your head is to think of a highly efficient molecular assembly line, where simple, common parts get meticulously pieced together into a complex and vital final product. Okay, so the assembly line starts up with some really basic raw materials. For the first 3 steps, the cell just grabs some simple molecules it has lying around, like bicarbonate and a couple of amino acids, glutamine and aspartate, and it starts putting them together. The whole goal, right here at the beginning, is to build that foundational 6-sided ring that forms the core of every single pyrimidine. And this diagram shows us exactly how it all goes down. First, carbamoyl phosphate gets built. Next, it's combined with aspartate, and then this right here is the critical 3rd step. The molecule literally folds in on itself, closing the loop to create something called dihydroerotic acid. And boom, just like that, our foundational ring is built. All right, so we've got our basic ring structure, but it's not quite ready for prime time yet. Now the cell has to customize it. So in the next 3 steps, it performs a series of super precise modifications. It oxidizes the ring, it bolts on a ribose sugar, and finally, it clips off a little carboxyl group. These are the finishing touches that create the very first usable pyrimidine. And what do you get after all that work? You get this molecule right here, uridine monophosphate, or UMP for short. Now, this is the key takeaway. If you remember one thing, it's that UMP is the parent molecule. Every single other pyrimidine the body makes starts right here. It's the central hub for the whole operation. So if we look at our diagram again, we can actually trace this exact transformation. We start with that ring we just built, the dihydroerotic acid. We can see it get oxidized, and then it goes through those final tweaks to become UMP. It's just a beautifully clear progression from a simple shape to a fully functional nucleotide. But wait, the cell's not done yet. It needs to supercharge these molecules so they're ready to build RNA. So in the final couple of steps, it phosphorylates UMP, which is just a fancy way of saying it adds 2 high-energy phosphate groups to create UTP. Then some of that UTP gets aminated. It swaps out a chemical group to create our other final product, CTP. And right here on the diagram, we see the end of the line. UMP gets those 2 extra phosphate groups becoming UTP. Then a quick little switcheroo transforms it into CTP. And with that, the assembly line is complete. The cell now has the fully formed, ready-to-go letters it needs to write its genetic story. Okay, so we have this amazing production line, right? But you can't just let a system this important run wild 24/7. That would be a huge waste of energy and resources. So the cell needs a really sophisticated control system to keep everything in perfect balance.
And one of the ways it stays so efficient is, well, it's just brilliant. Inside our cells, the first 3 enzymes that run this pathway aren't just floating around randomly. Nope. They're actually physically clustered together. You can imagine workers on an assembly line standing shoulder to shoulder, just handing the product directly to the next person. It makes the process incredibly fast and cuts out any wasted steps.
And there's another layer of control too. It's basically a simple feedback loop. Think of it like a thermostat in your house. When the cell has plenty of energy, lots of ATP, that's the go signal, turn up the heat. But when the final products, UTP and CTP, start to pile up, they act as a stop signal. They literally go back to the first enzyme and tell it to slow down. It's a perfect example of supply and demand at the molecular level.
This elegant little feedback loop is absolutely essential for maintaining that perfect balance, what biologists call cellular homeostasis. It guarantees the cell makes exactly what it needs, precisely when it needs it, no more, no less. It's just a system of profound efficiency.
Now, all this might sound pretty academic, right? Just a bunch of molecules and enzymes. But understanding this pathway is absolutely critical, because when this intricate molecular machine breaks down, the consequences for human health can be really severe. This is where biochemistry meets the real world. A classic example of this is a condition called orotic aciduria. It's a rare genetic disorder where that assembly line we've been talking about gets jammed up right near the end. If the body can't produce enough pyrimidines, cells can't divide properly, and that leads to some very serious symptoms like developmental delays and a specific kind of anemia. And what's amazing is we can pinpoint exactly where the breakdown happens. Depending on the type, one or even two specific enzymes in the line are defective. In both cases, though, the whole assembly line grinds to a halt right before UMP can be made. And this causes the precursor molecule, orotic acid, to build up. It's like raw materials piling up next to a broken machine on the factory floor. But here's the beautiful part. This is where understanding the biochemistry pays off. If the cell's own factory is broken and it can't make its own pyrimidines, well, we can just give them to the patient directly. By supplying them with uridine, we completely bypass the metabolic block. We give the cells the finished product they needed all along. It's a simple, direct, and effective treatment that comes straight from our knowledge of this pathway.
So, we can fix a broken pathway. But what if we wanted to break it on purpose? That's exactly what we do in cancer therapy. See, cancer cells are defined by their rapid, out-of-control division. And to do that, they need a massive supply of new DNA, which means their pyrimidine assembly line is running on overdrive. So drugs like 5-fluorouracil are designed to look like a pyrimidine, but they're fakes. They're like a faulty part that gets into the machinery and just jams the whole thing up, starving the cancer cells of the building blocks they desperately need to replicate.
And that really brings us to a much bigger, more profound idea. This pathway, it's just one of countless intricate molecular machines operating inside of us at all times. The more we learn to map these systems, the more we unlock an entirely new frontier of medicine, one where we can precisely target the fundamental processes of life itself. The question really isn't if we can do it anymore, but rather, what incredible discoveries will we make next?